Hello, my name is Ari,
My name is Noor.
and we are both undergraduates working on this project together in the González lab. We are building a photographic reference library for modern arthropods associated with fossil midden sites sampled in the Atacama Desert in Chile. Ladari Perez, a PhD student from our lab, is leading the study about the long-term responses of arthropods, and our project aims to help collect data for the study.
Arthropods are known to be the most diverse and abundant group of species on Earth, playing essential roles in ecosystem functioning, particularly in desert ecosystems. Despite their ecological importance and sensitivity to climate change, long-term data documenting arthropod responses to past environmental shifts is scarce.
In the Atacama Desert in Chile, fossilized rodent middens offer a unique opportunity to study preserved arthropod remains, providing valuable archives of past biodiversity and environmental conditions. This research aims to leverage these natural archives to investigate how arthropod diversity and community composition have changed over the past 50,000 years.
By analyzing these long-term records, we seek to improve our understanding of how desert arthropod communities respond to climatic variability. Additionally, we aim to develop a comprehensive photographic library to facilitate species identification and enhance predictions of desert ecosystem resilience under future climate scenarios.
Climate change is a major driver of ecological change, impacting species distributions, community composition, and ecosystem processes worldwide. Arid regions such as the Atacama Desert in Chile are particularly sensitive to climate fluctuations due to their extreme environmental conditions and specialized biota. Figure 1A shows the different arthropod hotspots on Earth. Figure 1B shows the 10 sites along a latitudinal gradient in the Atacama Desert, Chile, that our study focuses on.
Research investigates the long-term impacts of climate change on arthropod communities along a latitudinal gradient in the Atacama Desert. To assess these impacts over paleoecological timescales, we use fossil and modern rodent middens, natural archives that preserve biological material within crystallized urine deposits and rock crevices. These middens contain well-preserved plants and animal remains, including arthropods, providing a unique window into the past ecosystems spanning the last 50,000 years.
By analyzing both paleo and contemporary middens, we aim to reconstruct historical patterns of arthropod diversity, abundance, and phenotypic rate variation in response to past climate variability. This approach will allow us to better understand how arthropod assemblages have responded to environmental change over millennia and to develop hypotheses about the resilience and potential shifts under climate change scenarios in desert ecosystems.
So, our aim for this project was to develop a photographic library of collected arthropod specimens so we can support the accurate identification and classification of modern taxa, thereby enhancing the identification of fossil arthropods preserved in rodent middens.
PhD researcher Oderi Perez conducted arthropod sampling in the Atacama Desert using a combination of pitfall and sticky traps, strategically deployed based on habitat type, sticky traps in rocky terrain, and pitfall traps in sandy areas. Sampling we carried out across 10 sites and with 12 traps per site, each left in place for a 7-day grace period. Figure 1A shows an arid region where middens are usually found in rock crevices. These crevices are home to midden agents like rodents, shown in Figure 1B. Figure 1C shows an extracted midden.
Figure 1D shows a midden, which is a rock held together by the crystallized urine of the midden agent and has the biological and mineral material preserved in it. Figure 1E shows a sticky trap, and Figure 1F shows a pitfall trap used in sandy areas near the middens. In areas of high arthropod activity, traps were checked and replaced daily to prevent oversaturation.
Collected arthropods were sorted based on their association with paleo and modern middens, and subsequently processed for photographic documentation and further analysis. Our contribution in this project focused on photographing the specimens collected via sticky traps and organizing the resulting data to facilitate further identification and classification of arthropods.
The arthropods collected by sticky traps were preserved in ethanol prior to imaging to maintain their integrity. Figure 3 shows how we photographed these images. We used a Zeiss microscope equipped with an Ixiocam camera and Zen 3.8 software on a desktop computer to capture both general overviews and detailed images of these specimens. A key challenge during this process was distinguishing small arthropods from debris, particularly when working with the smallest specimens.
The most frequent encountered taxa included flies, ants, spiders, and beetles. To support accurate identification and classification, a size scale was included in each image. In total, we captured over 3,000 photographs, creating a comprehensive visual archive for ongoing taxonomical and ecological analysis.
Each photograph was systematically labeled with its corresponding site ID and Midden type to ensure tracking and future reference. These are our results. In Figure 4, we see some examples of the images we captured. The yellow background visible in the photographs is the sticky trap of the surface, the surface notably retained its adhesive properties even after prolonged submersion in ethanol, aiding in specimen positioning during imaging.
Photographs were taken at varying magnifications, adjusted as needed to achieve optimal clarity and detail for each arthropod specimen. This approach allowed us to document both overall morphology and finer taxonomic features crucial for subsequent identification and classification.
For our next steps, our future efforts will focus on the detailed identification and taxonomic classification of the photographed specimens, contributing to a comprehensive database linking modern arthropod assemblages to paleoecological records.
This photographic library will serve as a reference for comparing fossilized arthropods preserved in ancient middens. Integration of morphological data with genetic analysis could further enhance species resolution and provide insights into evolutionary and ecological responses to long-term climate change in arid ecosystems.
We are grateful to Dr. Gonzalez and the Gonzalez lab members, especially Udari, for their help in carrying out this project. Thank you so much for listening.
Thank you.